This video will zoom in and look at basic chemistry and accuracy of DNA polymerase III. In the previous video, we noted that DNA polymerase III only synthesizes new DNA in the 5' to 3' direction. And perhaps you asked yourself, why? The answer to that question boils down to the chemistry and the shape of the enzyme. DNA polymerase works by forming a covalent bond between the innermost phosphate group on the incoming nucleotide and the 3' OH group on the previous nucleotide. This enzyme can only synthesize DNA by forming this specific bond, and this is why it only works in one direction. DNA polymerase III is what is called a processive enzyme, meaning it can catalyze multiple reactions in a single binding event, which in this case is synthesizing multiple nucleotides into the new daughter strand. This is a complex enzyme, often called a holoenzyme, made up of multiple subunits that have various functions. We won't dig too much into this here, but one subunit, the beta subunit, is particularly important. The beta subunit forms a ring around the template DNA and is termed the clamp protein. This subunit is important for the processive nature of the enzyme and results in both higher rate and length of daughter strand synthesis during a single binding event. One of the key important features of DNA polymerase III is its fidelity. It rarely makes mistakes, which we appreciate a lot because low fidelity would result in tons of complications in our reproduction and just basic multicellularity. There are three main reasons for the high fidelity, which we will dig into here. They are stability of base pairing, structure of the enzyme active site, and the proofreading function of the enzyme. Complementary base pairs are the most stable form for the DNA double helix. Thus, correct base pairing results in the most stability of the newly synthesized DNA molecule. This stability reduces our error rate to about 1 error per 1,000 nucleotides. The enzyme active site further improves our error rate as improper base pairings result in a distortion of the helix that does not fit properly at the enzyme active site. This further reduces our error rate to somewhere between 1 in 100,000 to 1 error in 1 million bases. Lastly, DNA polymerase III has a proofreading mechanism that catches and fixes improper base pairings. When a mismatch is detected, DNA synthesis pauses while an exonuclease digests the newly made strand to get rid of the improper pairing. After this, DNA synthesis resumes in the 5' to 3' direction. Taken together, these three measures ensure an error rate of only about 1 error per 100 million base pairs at the worst. However, this would still be about 10 mutations each time your cells undergo DNA replication.